All of these discoveries are done by people. It all started when curious young students decided to become scientists. And we need more. We realized this long ago. There is talent and passion for science everywhere. We connect curious students with passionate young scientists. Now we are all together as Science Clubs International. Our common language is STEM. Our mission is to spark a social transformation by expanding access to high-quality STEM education and international scientific networks. We are growing. Empowering youth with science.
Okay. Hello, everyone. Good afternoon or good evening. My name is Chen Xue, and I'm a postdoc from Rogelio Hernandez Lopez's lab at Stanford University. So today, on behalf of Science Clubs International, I would like to welcome you to this webinar entitled Unnatural Selection, Resistance to Cancer Therapies. I would like to extend a warm welcome to our guest speakers, Dr. Subhadeep Dutta, Senior Scientist in Discovery Pharmaceutical Sciences at Merck, and Dr. Elias Vlahos, Postdoctoral Fellow at the Department of Chemical Engineering at Stanford University. This event is being streamed live on Science Clubs International's Facebook and YouTube, and we thank you all for joining us. So now I will hand over the microphone to our guest speakers for their introduction. So maybe we can start with Dr. Subhadeep Dutta.
Hello. Hello, everyone. First of all, thank you, Science Club International, for giving this platform to students. And I'd love to talk about a little bit about my career. So I started my career as a chemist back in India, where I did my bachelor's and master's. And I did a little bit of research on organic chemistry, to be specific. And then after that, I transitioned into United States for my graduate research or for my PhD. Here, I changed my stream a little bit, where my prior education was basic chemistry, but rather here I started exploring on the application side of the chemistry, where I worked in a chemical engineering lab where I was primarily working on different kind of polymeric biomaterials for various biomedical applications, starting from radiation sensing to endoscopic imaging or various bioprocessing applications as well. So following that, I had worked for 2 years in a gene therapy startup called Saliogen Therapeutics here in Massachusetts, where I was primarily working with lipid nanoparticle based technologies to treat a rare genetic disease for eye. Following that, very recently, I have joined the pharmaceutical company Merck, where I'm in the Discovery Pharmaceutical Sciences division, where basically we interface between the chemistry and biology group, which is in the discovery. When, for example, a lead molecule gets selected, that's when it come into our department for formulation development. And we support all the way to preclinical development, which means before going into human studies, we had to do a lot of animal studies and whatnot. But in general, we worked with a lot of different kind of modalities, starting from small molecule, peptides, proteins, different type of therapeutic applications. Uh, Also, I'd like to add that all of our discussion or my inputs today, it's primarily based on my learnings throughout these years, does not directly correlate any of my current research work at Merck. But happy to discuss overall topic. Glad to be here.
Great, thank you. Alex?
Yeah, so thanks for having me. It's actually funny. I think, Chen, we may have met at some point in one of the in Rogelio's sorting experiments. But I'm currently a postdoctoral fellow with Xiaojing Gao at Stanford in chemical engineering, and so my journey initially started in Canada. I'm Canadian, and I did my bachelor's in biochemistry. Really focused on sort of structural biochemistry and X-ray crystallography. And then I kind of shifted towards something more application heavy during my doctoral studies in the University of Toronto with Michael Sefton. And I was predominantly focused on developing strategies to transplant pancreatic islets underneath the skin. And so those are cells that help regulate your blood glucose, and it's a way to essentially treat type 1 diabetics. And then I shifted gears a bit into synthetic biology for my postdoctoral studies with Xiaojing Gao, where we've really been focusing on a class of circuits known as protein circuits to program cell behavior. And bringing this all together, I'm actually starting my lab at Georgia Tech in the new year as an assistant professor in BME, looking to sort of synergize these two ideas of tissue engineering and programmable cell engineering, to improve some health outcomes in the future. So my expertise is not on cancer, but a lot of these things are often interrelated. And you would be surprised what you could learn from one field to another.
Awesome. Thank you, Alex, and congratulations for the job. Okay, so let's start with our questions. Okay, so since this is a cancer-based webinar, I will ask the first question. What are a few definitions of cancer? And then why it's not correct to think about cancer as a single disease?
Do you want to start it off, or should I? Yeah, okay, you can start it off.
Sure. I think all of us kind of know cancer in a basic definition as, when you have a uncontrolled growth somewhere in the body. And to be honest, I think at this point, we realize that it can start from anywhere in the human body. I think in a cellular process, we all know that cells divides, it gets die at some point, but then if the process goes beyond the control, where it gets divided faster, or it uncontrollable growth and spreads other part of the body where it was not supposed to be there, and that's when you get some kind of weird symptoms, and that's when we start worrying if that is a cancer or not. But I think I would let Alex to shed more light or more color.
Yeah, I think it's tricky too, because when we think about cancers, there isn't really any one cancer. I think what Subhadeep really summarized nicely is it really is this idea of this dysregulation and uncontrollable replication. But I think the tricky thing is that although a cell has a lot of different fail safes, so ways that they event something goes wrong, the cell may undergo a process called apoptosis, which it's like sacrificing itself for the better good of the rest of the body. But there's a lot of these mechanisms designed to protect us from this uncontrolled cell growth, and cancer really happens when any of these fail safes are overcome. And so I think that's why it's not useful to think of cancer as a single disease, but more of potentially the ways in how cells become cancerous. There might be some similarities between different cell types, but you can even look at the stats at some cancers, for example, are much harder to treat than others. So if there was, if they were all treated the same way, you would expect your 5-year survival rate to be the same. You'd expect the modalities of treatment to be the same, and that's really not the case. It really comes down to how the different characteristics of the cells too, right? Like, depends what cell becomes cancerous, and some cells are a little bit more aggressive and others not so much.
Thank you. Okay, so let's go to the next question. What are a few drivers of cancer?
So some drivers of cancer, you can think of them as sort of like the way I look at it is like two steps. So you have oncogenes, which are cancer causing genes. And then you have tumor suppressor genes, which are sort of like, think of the brakes of a car. And so the way that, cancer can occur is that if you have in one case, you have like a mutation in these oncogenes so that they're like, always turned on. Your car is going nonstop and it's really just going fast, and that's not ideal. And then, if you have a deletion or a mutation in some of these tumor suppressor genes, then what you're effectively doing is the cell is getting rid of the brakes for that car. And so you're kind of getting the same effect where you're just getting uncontrolled speed, but in two very different ways. And these two sort of, I guess, pathways are pretty are redundant, but they're also interconnected, and that's more on the different drivers. But I think Subhadeep can talk about specifically what can cause cancer, let's say UV light or etc.
Yeah, I think Alex gave a really good real life explanation or a scenario to cross connect how the scenario of the car relates to our body. Just to compare the perspectives. But I think as he has generalized, what are the drivers, right? Like the two different type of genes, but specifically, like there are some external stimuli which basically led the genes to be behaving differently, right? Like those external stimuli could be a variety of different kind of stuff, starting from physical and chemical agents, like when someone is smoking too much, or if someone is exposed to UV for long amount of time, right? So these are external stimuli which could essentially make changes in the body. And there could be other aspect. For example, there are many reports of different lifestyle factors, such as diets or consumption of alcohol. Again, there are, I believe, lot of streamlined studies still need to be done to correctly understand how each of these factors, how aggressively they actually are the drivers of the cancer. Because if you think about it, we are like, I was in Arizona, which is, we call a sun state, where we have a very large extent of sunlight. And as you can imagine, in very hot summer, where you could essentially get a lot of UV rays, even from sunlight and other sources. So what I want to say is that there are multiple level of factors, starting from radiation or other kind of infection or inflammation, could essentially ties back to the disease. But I believe it's very difficult. We live in a complex world with different kind of atmosphere, and scenarios. So it's very difficult to pick one cause to say that actually caused the cancer. So that's why I feel it's still a complex problem. And essentially, it starts from the detection, and we start to look at where it happens and how it's progressing. So that's what I would add.
Great. I think that's very comprehensive answers. So we can move to the next question. Okay, so what are the stages of cancer and what do they mean?
Yeah, I think as I was saying previously, is that I think the cancer definitely starts with the detection. And as I was initially telling, that it's abnormal growth in the body. But sometimes, as you can imagine, different organs are in different parts of your body. And sometimes it's easier to notice the growth quickly based on where that growth is in the body, versus if it's in a organ which is not so visible, or we don't really observe that closely, then we might miss that small increase of the organ, or small growth of the organ, and we may find out much later. So, and also it depends on, as Alex was saying, different cells grows differently. So how a cell in our hair grows versus a cells in our hand grows is much different, right? So that also plays a huge role. In general, I would say that the stages usually refers by numbers, like stage 0, 1, 2, or 4. But essentially it's basically telling you how aggressive those stages are. So 0 means it's basically very early stage, but the 4 is very large later stage, where it may have progressed into the other organs. But Alex, feel free to jump in.
Yeah, so I think the simplest way I like to think of it is, I think there's technically 5 stages if you consider stage 0, but there there's stages of cancer. The higher the number, the worse prognosis you are. And I think, there are some nuances depending, between stages 2, 3, 4, etc. But really it's sort of to what somebody said, is it's whether or not that initial cancer mass has spread to other tissues, and to what degree have they spread. So you can think of sort of your body is completely interconnected with each other. Some of these systems, we think of the human body as different systems. So you have like a circulatory system, you have a lymphatic system, you have a nervous system. And so what really happens is, the beauty of our bodies being so interconnected is that, information can be passed from one part to another very quickly. But this also you can think of it as like a highway, so that if this car that is going way too fast can now, be found somewhere else as well. And some of these, cancers, depending on what organ or site they start from, have a tendency to sort of spread more than others. So something like pancreatic cancer, where I know a little bit more information about that because of, individuals, my work has been on diabetes. But one tricky thing with the pancreas is the pancreas, one of its functions is to secrete hormones that get sent directly into the bloodstream and go throughout the entire body. And pancreatic cancer is one of the sort of the worst types of cancers you can be diagnosed with, for 2 main reasons. One is that it's very hard to detect. So like Subhadeep is saying, like if it's hard to detect, by the time you have, you develop symptoms, it's kind of probably in its stage 3 or stage 4. And then the other tricky thing is, because it's so interconnected to the circulatory system, it's very, conceivably, it's very easy that it can metastasize. And metastasize, metastasis essentially is just when you have this the initial cellular mass or cancerous mass being spread to other parts of the body. And that's where it becomes really hard to treat, because when a cancer has become stage 4 or metastatic, you really need to try to ablate as much as possible, because if even a small portion remains, you'll go into remission and you won't have symptoms anymore, but eventually the cancer will return. which is why something like pancreatic cancer has such a poor survival rate, because you can treat it as best as possible, but it's very hard to treat everything.
Okay.
Yeah, I think that's why I think I would like to add that we all think about the how we can treat cancer, but as Alex was saying, like some of these detections are being made very late stage. So I think there are tremendous amount of research right now is going on how to improve this detection, how we can detect this cancer in very early stage. That's where it can be still treated. But if we finally get to detect the cancer in third or fourth stages, that's when it becomes really tough and you can hardly cure that cancer.
Thank you. Okay, so for our next question, I think you guys already talked about this a little bit. So do you know about cancer diagnosis? What tests are routinely used?
This is okay. Cancer detection tests. So some of the cancer detection tests have to sort of deal with specific biomarkers. So in some cases, so like let's say prostate cancer, there are certain proteins that if a cancer cell is in your, I guess if the prostate has become cancerous, they're going to overexpress and start replicating nonstop. And then there are markers associated with, this uncontrolled growth of, let's say, prostate cells. And there's known as, one of them is the PCA antigen. And so what one could do is you could actually sample the blood of an individual and run an assay known as an ELISA. And an ELISA just uses antibodies specific to that given protein. And what you can do is then you can quantify the amount of that given protein in a human. And if it's above a certain level, typically that would suggest that it's abnormal, and then you should do some further testing to validate whether or not an individual has that given cancer. But to I think Subhadeep really talked about the detection. It's you'll have some of these potentially quick and dirty tests for detection, and then you kind of have to do some follow-up tests, because you want to make sure that you have a real diagnosis before doing some of these treatments, because they can be quite, some of these treatments can be quite harsh.
Yeah, I think it's a great introduction, and what Alex was saying, I think that's one of the newer ways, and especially like cancer, like prostate cancer, also one of those aggressive cancer. If you have like in late stage, then it's very hard to kind of cure that. But I think having those biomarker based tests are very essential, which we have now. But I think if you think about it, as I started talking about the cancer, about the abnormal growth, I think in general, very early days, decades back, it used to be like a biopsy, right? And it still does now, but I think it does with some modern test like imaging test. I can talk about a little bit about that. I have worked some of those aspects in my PhD work. But one of the thing, is just biopsy, which biopsy technically means is that you are collecting the tissue from those organs where you think that it could be a cancer, which is like a lump or kind of uncontrolled tissue growth, and you are literally looking under the microscope. Sometimes you stain with different kind of markers to identify the bad cells, but those are very traditional ways. But the other aspects were also the imaging modalities, and this is where I would say some of these techniques are invasive, and some of these techniques are highly costly as well. And that's where the challenges comes. Many of the countries where they do not have good access to these type of techniques, like for example CT scan or X-ray or MRI. These are complementary techniques. CT scan and X-ray kind of tells you the contrast between your bones versus your tissues, but MRI tells your tissue morphology more than the bones. So basically these two are complementary techniques. Most of the cases patients gets both of these to find out a certain kind of cancer. But the other aspect, especially if the cancer happens in the GI tract or anywhere close to the GI tract or the gastrointestinal tract, that's when people do endoscopy. And that is one of the, I would say, one of the invasive techniques which someone has to go through. It's very painful, but as Alex was saying, it's not one time thing and it's done, right? Because essentially you do one time, you find out something is okay or not, but you do have to go back and do that maybe in few months to find out if that tissue has changed or if that has an extra growth or not. So that's where I think there are a lot of new techniques and modalities needs to come, which are less costly and also non-invasive.
Great, thank you. So now we're going to shift gear from cancer detection to cancer therapy. So the question is, I think we still have the first question to answer, which is what are some therapeutic approaches against cancer?
Sure. I think I can start a little bit as I was talking to the biopsy part. So, as you can imagine, right? So, you found an abnormal growth in the tissue. So, the very old school way of treating that would be just getting rid of it, which is basically means a surgery. But the problem happens when that tissue is in a sensitive organ, right? Or as Alex was saying, if that has metastasized, which means that has gone to the other parts of the body. So that's when some of those newer approaches came in. For example, uh, I have worked on also the aspect of radiation therapy. I can talk a little bit about that. And I would say nowadays it's becoming more and more sophisticated, where it can actually treat cancers or relatively cure or maximize the survival rate of a brain cancer. So, and most of these techniques has lot of relatively quite a bit of side effect, but it has able to decrease its side effects quite a bit based on its sophistication on the technical aspect. So earlier, radiation therapy is basically a therapy where you are giving high energy to a certain place of the body to kill those cells which has been growing abnormally, right? But you can also think that by doing that, you maybe will be hitting some of the healthy cells which are around this abnormal growth, right? And that's when you may see some kind of side effect. And the real improvement of these radiation techniques nowadays is that there are, as I mentioned, different kind of imaging modalities has gone into, and nowadays all of these radiation therapies are pretty much computerized, which means there are different imaging techniques is being done on the patient to get a 3D recollection of that whole organ. Now the doctor can come in and see, okay, this is where the tumor is, this is where the abnormal growth is, and after that, the radiation beam, which is the machine to kill those cells with those high energy, it can really focus and pinpoint those certain small parts of the body. And the good thing about these techniques nowadays is a more sophisticated way. They call it fractionated radiotherapy, where those with very smaller dose, but they repeat for longer times or in shorter intervals. That's when you get the total dose, but over many weeks. So this is how you basically treat even a very complicated, complex organs like a brain.
Yeah, and sort of really the name of the game with this is trying to minimize as much damage to healthy tissues as possible. And then that also sort of goes into why it's so challenging with the further stage cancers, where if it's relatively local, it can be at times pretty straightforward on what needs to be done, whether surgery is feasible or if you do your radiation in a very specific part. But when it gets tricky is when it's all over the place, and then that's when you'll have to use some sort of chemotherapeutic. And the tricky thing with cancers is that a lot of the current modalities are non-super specific, because cancer is just a dysregulation of a cell state. And so when that ends up happening, it's tricky, because you hear about all these stories of being on chemo, and often you see these side effects, and a lot of the side effects are often because cells that replicate quickly, like cells that line your gut, also are affected really by these chemotherapeutic agents, because that's what a lot of these sort of agents are designed to do. It's really try to arrest cell growth as much as possible, or target these like really quickly replicating cells. And so one therapeutic approach we haven't really talked about is like these CAR T, which is essentially taking a T cell, which is a native cell within your body, that is designed to target other cells for destruction. So they're part of your normal immune system. But what's kind of pretty cool is that you can actually engineer them so that they become specific. They target a specific protein. And so some of the work that Rogelio is doing in his current lab is trying to improve these therapeutic cells, or these CAR T cells, in order for them to be safer and really improve their specificity, right? Because CAR Ts also have a very similar issue, where you need to make sure that you're targeting something that's specific to cancer, which kind of comes back into this whole biomarker thing. But finding those specific proteins and biomarkers is the tricky thing. But there are cool ways that one could do to sort of like improve specificity by imparting some logic or programming capabilities into CARs, for example, to sort of just have them only respond when you have multiple proteins being present, to offer a little bit more specificity. And I know that there's a huge push right now into CAR Ts, as well as CAR NK, which is a natural killer cell, which is just another type of immune cell.
Okay, thank you. Okay, I think for the next question, it's kind of overlapping. So please describe the general mechanism of approaches like chemotherapy, biologics, cellular therapy against cancer. So do you have anything to add on to it?
So, okay, I guess I kind of talked a little bit about it, but part of it is so with something like a small molecule, you can try to target specific pathways. So there's an oncogene known as RAS, and RAS is one of like the most, that RAS signaling pathway is one of the ones that is mutated often in like pancreatic cancer cells. And so there's different types of mutations that can occur. And one of the ones that can occur is by changing, I think, so the glycine residue on 13 to cysteine, which is just another type of amino acid. And what happens is when you have that mutation, the RAS signaling pathway just activates nonstop. And RAS is involved in a lot of cell differentiation and replication, and it pretty much just turns the cell on nonstop. But that mutation can also occur to other amino acids that effectively act the same way. But what people have done is they've developed small molecules that can target cysteine, because cysteine is a unique amino acid that it has a thiol group, which is a sulfur, I guess, functional group, and you can actually leverage that to sort of target, design small molecules against that. So there are some small molecules that are a little bit more specific, but then again, it's the tricky thing with small molecules is that inherently, if they're going to be approved for clinic, they have to be very, very specific as best as possible. Or else you're going to have a lot of off-target effects, and that's not ideal. And then do you want to talk about chemotherapy use, and then I can come back to cellular therapies?
Yeah, I was thinking to also bring the aspect of hormonal therapy. I think that is typically relatively new, I would say. As you can imagine, it names with a hormone. So basically, it can treat cancer which uses any hormone to grow, right? Like, for example, breast and prostate. These two cancer type has certain hormones which makes these hormonal growth, or the abnormal growth of these two type of cancers. So these, like, the therapies like hormone therapy, typically they use it, I would say, right now in combination. For example, either a patient gets a surgery or a chemotherapy, or a radiotherapy, and on top of that, they get occasional hormonal therapy to kind of arrest any further growth, which basically blocks the hormonal pathway. So, I think Alex talked about the small molecule aspect, but I would say that chemotherapy essentially is one name, but a lot of different kind of drug modalities falls under the chemotherapy domain, and which includes small molecule, but it could be other things like a peptide, um, or so. But I would say that the main thing is always about the chemotherapy specifically is to kind of minimize the off-target effect, especially if it's a small molecule where it can go. Essentially, you are giving within maybe injection most of the cases, or an oral pill, but it can go anywhere in the body where it can have this off-target effect. How to minimize that? But feel free to chat about the cellular therapy, Alex.
Yeah, and so the cellular therapies, I guess, predominantly would be like the CAR T, which I talked about, which it really tries to leverage the natural killing potential of T cells, so that they are specifically killing the cancer cells based on what they're expressing on the surface. So the way that these um, cancer cells are recognized is about, the T cell will recognize a specific protein, and the their receptor that they're engineered to is designed to be against a specific protein. And so when those T cells recognize that protein, they'll be activated to kill the cell. And it's through the natural, I can't remember the exact pathway, but they'll secrete some ligands that essentially create little holes in the cell membrane, like perforin and granzyme, I believe. And then there are also other, mediated cell killing, cell specific killing things as well. But really, what you have to do is engineer the cell first, and then from there, that's where you end up getting the killing activity of the CAR Ts.
Great, thanks. So our next question is for cancer vaccine. So what makes it challenging to develop a cancer vaccine?
I can start a little bit on some of the perspective, and I think I'm not sure how much we know about it, but there are quite a few of companies who are doing clinical trials for cancer vaccines. Now, I think the challenges comes from the fact that we don't completely understand this disease, right? It's so complex. It can progress in very different ways, where it starts to where it goes. So it's not a straightforward disease where you only have maybe one pathway. Most sometimes there are two or three pathways, are dependent on each other. And our body is also talking to each other. So main hurdle is basically try to understand the disease. If we really understand the disease, that's when it's much more easier to design something as a vaccine. Now, in recent times, as you know, there are a lot of progress in mRNA therapeutics or mRNA vaccines. Essentially, what that is that, as you know, proteins, as Alex was saying, most of the time, when this cancer happens, there is some protein which gets upregulated or basically produced more and more, right, than it's what it's needed, or downregulated, either way, right? Like some proteins are dependent on other. So you can say one protein is upregulating, other protein is downregulating, and whatnot. But if you go a little bit back to that, right, protein gets produced from the mRNA, right? That's when we call it, say, messenger RNA. Essentially, that's the code to the cells. And the mRNA, when it goes into the endosomal escape, that's when it makes or produces that protein inside the cells. Now, the best way would be to actually stop that protein production, if that is over regulating, or if it is producing more, or the other way is if it's down regulating, it will be to produce that more, right? So that's what the mRNA plays a role there. Now, because of this new technique or invention, what we saw because of the COVID, although there were 2 decades of research was going on to develop mRNA therapeutics and vaccines, but there was no certain disease which was very prominent to us and easy to solve. And that's where COVID happened, and there were a lot of development where there was a new type of vaccine, which is basically mRNA vaccine, which came up. And primarily that is made up with technology called lipid nanoparticle. So lipid nanoparticle is basically a vehicle, right? When someone delivers a package to your home, think about that package as the mRNA. And but the mRNA is not stable inside the bloodstream, right? So we cannot really inject mRNA inside our bloodstream. So that's when you need a package or a box where that box actually can go into the cell and then release, right? So when you get a package, it comes into a box, and you get the box and then you open it. So that's what the a lipid nanoparticle technology does. So when we inject a lipid nanoparticle based mRNA vaccine, it essentially goes intravenously, but it goes all the way to that targeted cells. And again, that aspect of targeted therapy, what Alex talked about, to reduce the off-target effect, that also is very important. Otherwise, it will end up into the unwanted places where you don't want it to go. And that's where I would say there's a lot of this novel approach where lipid nanoparticle based mRNA vaccines are being produced by some of the companies or the industries which are working hard. And there are a lot of clinical trials currently happening on various kind of cancers currently. And think about it, as I mentioned, its clinical trials are happening, which means there were early success in animal models, models like mice or rats, or even larger animals like dogs or a monkey. That's when you get approvals from these organizations to actually do an human trial, a clinical trial in humans.
I think the other thing too is it, in a way, like the vaccine for cancer is kind of similar to the CAR T approach in the sense that you're trying to drive a very controlled and a controlled and programmable is not really the right word, but essentially a controlled immune response to a given cell. So sort of how Subhadeep was saying, where it's just like, you want, when you give it a vaccination for something, typically you're immunizing against a specific protein on a virus or a microbe that your immune system will recognize. And in this case, one tricky thing is, it comes down to, again, like, how do you find something that's very specific to the cancer cells? And then the second thing too is that cancer in general, the way it actually is able to replicate in the body is that it is able to overcome the immune system by creating immunosuppressive environments. And what that means is that immune system can try to destroy it, but there are mechanisms and proteins that are secreted to make the environment immunosuppressive. And it's, we kind of know the immune system normally, in its everyday function is actually designed to also get rid of cells that go awry, or get screwed up, because you have individuals, for example, who are on immunosuppression, chronic immunosuppression for something like a cell transplant, like an organ transplant. And often, one of the secondary or not complications, but potential side effects of being on systemic immunosuppression for a long period of time is you have a higher incidence of cancer. And part of that is because your immune system really is also designed, it's part of the fail-safe against these cancer cells, that, if something is going a little bit funky and the cell doesn't undergo apoptosis and kill itself, then sometimes you'll have the immune system come in and sort of just do it for it. So I think what makes it tricky for this vaccine for cancer is that you're trying to program the immune system for a very specific target. And with something like a normal vaccine, where whenever you get infected, it's something foreign and it's very, very different than a normal cell. Yeah, you don't need to maybe understand fully how you're developing an immune response against it, because it's so specific to something that only will be foreign. But cancer cells are not foreign, which makes it increasingly tricky.
Awesome. So I think here comes to our last question. So what are the new therapies for recurrent cancer? Or maybe I'll rephrase this question as, what do you think the future of cancer therapy will go?
Yeah, I think I'll kind of touch base on some of the aspects. But I would say that the therapy and the cell therapy, specifically, immunotherapy is a broader domain, right? Like, immunotherapy is definitely an emerging field, as I see in the questions. And we touched some aspect of the immunotherapy, right? Like, Alex gave a really good, I would say, crash course on immunotherapy, in very different, in various answers. But also I talked about the vaccine, right? But the other aspect could be monoclonal antibody or a cytokine based therapies. Those are also different ways of doing the immunotherapy. Cytokines, for example, it's a group of protein which naturally is in the body. Now, especially for various kind of blood cancers, that's when the cytokine based immunotherapies are getting used. And I would say that we classify these immunotherapies, different immunotherapies, based on what is the active drug to treat with that kind of immunotherapy, right? Monoclonal antibody is a specific type of molecule, which basically targets to cure that disease. That's when monoclonal antibody therapies is one type of immunotherapy. But the basis is the same, as Alex was mentioning, is essentially you are training your own immune cells to fight back to that cancer. Now, one of the aspect I think emphasize, and I'd like to reiterate, is that I think right now we feel very confident, fairly confident on the aspect, at least in you know countries where you have lot of availability of these kind of modern cutting edge cure to treat cancers. But again, it starts from the detection. So I do feel that there is a lot of improvements need to be done, and a lot of research is happening forefront to actually detect those rare cancer where it is actually getting detected in stage 3 or stage 4, which is basically highly critical stage, when it has been already metastasized, where it has been started growing in the other parts of the body. So I do believe that we are in the era where we will see a lot of innovation and more fundamental research to find out more ways to detect those hard cancers which are getting detected in late stages right now. So that's what I think right now would be more things coming up.
Thank you.
Yeah, I think I'll keep it short because I know we're running out of time, but I think it does ultimately come down to sort of, once we are able to detect these, we need to also be able to treat them. And I think a lot of the current mechanism or the current methods really need to have this specificity. And I think to Subhadeep's point too, is just like, if we had a very, good marker of a hard to treat cancer, you could leverage that both for detection and potentially treatment, right? And I think that's why there's such a big promise with the CAR T work, but I also, there are issues with CAR T as well. So I think, we need to acknowledge that specificity again seems to be the biggest issue. And there, even though we can engineer them as best as possible, there is still some potential off-target effects that they can occur. But I really fundamentally think that if we are able to come up with some really nice and specific targets, and whether or not that target is one that we use for detection, for therapeutics, or even both. I think an ideal target is one that you can use for both. I think that is still, we can come up with all the cool ways we want to kill cancer, and I think people have shown really cool ways to kill cancer in a dish. But I think then when you actually put it in the complexity of the human body, where there's so many different potential cells, but also pathways that we have to take into consideration, that's where the specificity fundamentally becomes one of our biggest. I wouldn't say it's our biggest roadblock, because another argument could be delivery, right? We have these potential modalities, but if you can't deliver them into the cancer cell, then it doesn't work. But yeah, I think there are some really nice work that's going on to finding specific biomarkers, and then also focusing on this delivery issue.
Great, thank you. So I think that's all the questions that we have. Let's see if there's any questions from the audience. I don't see any. So
it makes our life easier, so that's good. we were so good at explaining it. That must just be that. Yeah.
So yeah, thank you again, Subhadeep and Alex, for all these fascinating answers and inspiring answers. And then thank you all people for attending this webinar, and we'll see you next time. Thank you.
Yeah.
Bye.
Also, thanks Science Clubs International for having us. It's I think Subhadeep and I did our first class last year or the year before, and
2 years back, I believe. Yeah,
yeah,
it was
yeah,
a
and
good memory for sure.
yeah, no, it's good. So we're always happy to help in any way we can.
Definitely. Again, I'd like to conclude again, as Alex said, I think I do believe that we have our role to inspire the next generation of students who are thinking about going into this STEM education, right? But unfortunately, some cases probably they don't get to hear about these school sciences in those situations where they are. And I think these type of interactions only to inspire those minds to motivate those new generation of students to come into these emerging fields and make some progress.
All right. Thank you again.
Perfect. Thanks.
Bye-bye.
Bye.
Bye.
All of these discoveries are done by people. It all started when curious young students decided to become scientists. And we need more. We realized this long ago. There is talent and passion for science everywhere. We connect curious students with passionate young scientists. Now we are all together as Science Clubs International. Our common language is STEM. Our mission is to spark a social transformation by expanding access to high-quality STEM education and international scientific networks. We are growing. Empowering youth with science.